Okay, guys, today we're going to talk about the peripheral nervous system afferent division. This is just an introduction, and next time we're going to talk about start talking about special senses such as vision and then hearing, and then other special senses as well. That's going to be the end of our course. So this presentation is fairly short. I just mean to introduce here to several different concepts before we start our tour of special senses. First of all, today we're going to define conscious versus unconscious perception. We're going to define the general and special senses. We're going to also classify sensory neurons from different point of views. We can classify them by type, by stimulus detected, or by location. And then we're going to talk about sensory coding and all that's associated to it. So, of course, we are talking about afferent division here of the peripheral nervous system. And remember that this afferent division, sensory division, is subdivided in somatic sensory and visceral sensory. And in both of these subdivisions, we can recognize both general and special sensory functions. Okay, so now first of all, we have to understand that some sensory neurons travel to the cortex, and in that case we talk about conscious perception. Remember that most of your sensory functions travel through the hypothalamus, right? We know that that's the boss basically, many of the sensory functions and stimuli, they arrive to the cortex from there. And so you have a stimulus that is uptaken by a receptor here in the peripheral nervous system, and typically there are several neurons through which these stimulus actually travel before it arrives to the cortex. So if the actual stimulus arrives to the cortex, then we talk about conscious perception. Whereas if the stimulus does not travel to the cortex but pretty much ends at the thalamus, then we talk about unconscious perception. Okay, and of course you have the same thing going on. You have a sensory receptor or sensory neuron, and then a few neurons in between before that stimulus reaches the thalamus. And we can recognize several conscious types of stimuli as well as subconscious or unconscious. And within the conscious and subconscious as well, we can recognize the somatic stimuli or somatic senses. And in the conscious, we also recognize the special senses, okay? Whereas in the subconscious, we have the somatic stimuli and the visceral stimuli. So let's look at the conscious first, the conscious stimuli, or conscious senses, we can say. The conscious ones, as I told you, they include the special senses and the somatic senses. The special senses, which actually we're going to look quite in detail, are the vision, hearing, taste, smell, and equilibrium. These are all conscious. And among the general conscious somatic senses, there are touch, temperature, pain, itch, and proprioception, which we're going to defined as awareness of the position and movement of the body. Now, whenever the stimulus processing is subconscious, we can again distinguish between somatic stimuli and visceral stimuli. The somatic stimuli are the ones that refer to muscle length and tension, or as well as proprioception. Whereas, and see, proprioception is something, part of proprioception is really subconscious, whereas there is, some other part that is conscious. And regarding the visceral stimuli, the subconscious stimuli processing, we have things like blood pressure, distension of gastrointestinal tract, blood glucose concentration, internal body temperature, osmolarity of body fluids, the concentration of solutes in body fluids is maintained always in a certain range. So many of these fall into homeostatic pathways, which are controlled by visceral stimuli. Lung inflation, pH of several fluids such as blood, and also the cerebrospinal fluid. Okay, so there is a constant stimulus of all these factors, and if ranges of these factors go out of the normal or what it should be, then there are pathways that are activated through this stimuli that will reestablish those factors in the normal range. All right, so now we can actually subdivide the senses also as general senses and special senses, just in two broad categories, okay, without doing too much subdivision here, if you will, okay? So general and special. What are the special senses? There are vision, hearing, equilibrium, olfaction, and gustation. These are the special senses. You must remember this for me very well, okay? All the others are general senses, okay? So if you remember what are the special senses, you don't even have to memorize the general senses, because whatever it's not this is this, right? So some somatic, chemicals, temperature, pain, touch, proprioception, pressure, visceral, chemicals, temperature, pressure, okay? So again, this can be, there is pressure twice because this can be somatic or it can be visceral, okay? That's why sometimes it will list them twice for you, okay? So general senses, as we saw before, we can recognize those that are somatic and visceral as well, okay? All right, so we can look at things from different points of view. We can define them in a more broad way, or in a broader way, or in a more schematic, more subdivisions. But bottom line, you have to be able to just think about a certain sense, and it's pretty obvious if it's somatic or visceral, okay? Something like touch or pain, okay? These are all somatic, right? Whereas something like temperature, okay? Temperature in the sense of maintaining the internal body temperature, as we have seen, is visceral, okay? But sensing the external temperature is somatic, Okay, so you have to be able to distinguish these things. You have to think about it. If it's something that you can consciously um, consciously sense, then of course it refers to the somatic sense of temperature. But if it's referring to maintaining internal body temperature, this is a visceral stimulus, it's subconscious. Okay, it's pretty obvious. So please don't get confused between these different temperature stimuli processing, okay, and in which category they belong. All right, so okay, so with that said, you have to know very well what the special senses are. Okay, it's pretty easy: vision, hearing, and then gustation, olfaction, which is the sense of smell, and equilibrium. Okay, 1, 2, 3, 4, 5. You have to know very well for me. Okay, now we're going to talk about sensory receptors, which are obviously these cells that sense stimuli and initiate a response. Now, we can classify sensory receptors by type. There are different types. There are simple receptors, there are complex neuroreceptors, and special sense receptors. The simple ones are neurons with the free nerve endings. Simple as that. They're just neurons with the free nerve endings. Pretty simple. Then there are the complex ones. Usually these ones have nerve endings that are enclosed in connective tissue capsules as shown here. And in the next slide, I will recall, we have pretty much already talked about these two types when we were talking about skin, but I'm going to remind you about those ones the next slide. There are special sense receptors, which are cells, really, special cells as shown here, okay? They're separated, they're really specialized cells, okay? And they are able to release neurotransmitter, okay, in a cleft. That neurotransmitter will act on a sensory neuron, okay? And we'll see what happens whenever there is a stimulus. And as you can already infer, what's going to happen upon a stimulus is generation of an action potential. That's always the case. Now, the way this action potential is initiated can vary slightly, but ultimately, an action potential needs to be always initiated in order for a stimulus to be propagated. Okay, so here I wanted to remind you some simple receptors that we have looked at in the dermis and epidermis. Remember, those are the free nerve endings that we can find. The only type of neuronal cells, nerve cells that we can find in epidermis are simple free nerve endings. It's not shown here much, but there are some that go in here. Whereas things like the Meissner corpuscle, the Pacinian corpuscle, that I don't know if you remember, or we talked about this when we talked about dermis, and I showed you the Meissner corpuscle, if you remember, looks like a cotton candy. And this is a Pacinian corpuscle. These are some of the histologies that I actually presented to you, okay, about these receptors present in the dermis. And things like the Pacinian corpuscle, these are complex receptors. You see that they have a special capsule around them. Okay, so these are some examples. Now, besides the type, we can classify by type. We can classify sensory receptors by stimulus detected. Okay, you know that there are some receptors that will sense pain. Some receptors detect smell. Some receptors will be photoreceptors that are detecting light, and then and so on. So these are all the classes of Sensory receptors by stimulus detected. Mechanoreceptors, they will sense mechanic stimuli, so a touch, for instance. Thermoreceptors will sense change in temperature. Okay, chemoreceptors are able to detect chemical molecules. When you are smelling flowers, there are these molecules that are stimulating the chemoreceptors present in your nose. Photoreceptors detect light, okay, and nociceptors, they determine pain. And lastly, baroreceptors. Baroreceptors are pretty much mechanoreceptors, but they're located specifically in the carotid sinus and aortic arch, as shown here. Okay, there are receptors here in these big aortic branches of your body, and the function is to sense the pressure changes and by responding to change in the tension of the arterial wall. So they initiate this baroreflex mechanism, which is this fast response to changes in blood pressure. Okay, so they're able to sense the pressure changes and respond to change in tension of the arterial wall. Okay, so again, these are important. They are to be known. Okay, this is a classification of sensory receptors by stimulus detected. What kind of stimulus can be detected? And you know that these receptors are very specific. They can only sense a certain type of stimulus. The third type of classification of sensory receptors we can do is by location. By location in sense of somatic or visceral, okay? And so we can define somatic sensory receptors and visceral sensory receptors. Here is a list of somatic sensory receptors in the next two slides, 1 and 2, okay? What are the somatic sensory receptors which are already introduced? Temperature, pain, pressure, touch, these are all somatic. And these are all located pretty much in skin, dermis, epidermis, and respond to, they're separated, and specific receptors respond to specific stimuli. For instance, there are some dedicated to pressure, some dedicated to sensing temperature, pain, and so on. Then there are the receptors for proprioception, and these are specialized neurons which will sense joint position, tension, and level of muscle contraction. And they constantly relay a message to the brain, and they also interconnect with those pathways that are dedicated to maintaining a balance. These are especially in the inner ear. So, as a whole, these proprioceptors, they pretty much have an important role in maintaining body posture, also in some maintenance of equilibrium. Smell, taste, vision, equilibrium, and hearing are other examples of somatic senses. Okay, and here they are shown the specific sensory receptors associated with each of these sense. And we're going to look at them quite in detail. Okay, the next thing we're going to talk about is vision, and we're going to define all these receptors. Then we're going to talk about equilibrium and hearing, and then taste and smell. And of course, these particular receptors are located in particular organs. And for instance, the um, receptors for smell are hair-like, they present hair. These are known as cilia, are present in the nasal cavity, whereas the taste ones are present on the tongue. The vision ones, of course, are present in the eye, in the eyes, in the retina specifically, and equilibrium and hearing are present in the inner ear. Okay, the specific sense. Regarding the visceral sensory receptors, these are located all over the place in visceral organs, and these monitor internal conditions and the status of organ systems, and they initiate response whenever values or factors go out of homeostatic range. Now, what happens after a sensory signal is received, generally speaking? As we already said, most of the pathways, they travel through the thalamus, okay? And basically from there, some of them are actually sent to the primary somatic sensory cortex, the conscious ones. The subconscious ones, they stop at the thalamus, basically. And so most of the sensory pathways project to the thalamus, and the thalamus relays information to cortical centers. Now, when we talk about olfactory pathways, we already learned this. If you remember, the olfactory pathway does not travel through the thalamus. It travels directly to the olfactory cortex, okay, through specific pathways without traveling to the thalamus. The equilibrium pathways project to the cerebellum, so also don't go through the thalamus. So remember these two exceptions for me. Okay, and here I just wanted to give you an idea of the amount of space of the sensory cortex that are devoted to each body part, and how it is proportional to the sensitivity of that part. And of course, there are parts of our body there are bodies that are more sensitive than others, right? And that's because the amount of cortex devoted to those body parts is huge, okay? And of course, our genitalia, for instance, are this is kind of funny, right? Have lots of these. A lot of sensory cortex is devoted to those parts, the sensitivity of those parts, and face, tongue, okay, parts like these are also important. Also, we learn about hands and feet as well. Okay, well, we learned this already. Okay, so we talk about somatic pathways, okay. For instance, a sensation of pain, temperature. Of course, this travel through the spinal cord, and then through the thalamus, and again to the cortex. Okay, and the sensations are perceived in the primary somatic sensory cortex, okay? We already talked about all these when we talked about the cerebrum, right? Okay. And remember that between a primary sensory neuron, which is the one that really is responsible for picking up the signal, there are always interneurons basically between that and neurons, for instance, that are present in the thalamus or in the cortex. So there are secondary and tertiary neurons most of the time that actually create bridges. Okay, so there isn't just one neuron that receives a signal and directly transmits that information to the cortex. There are many interneurons, typically secondary sensory neurons and tertiary neurons as well. All right, now before we move on, I just want to make sure that we define terms here. So when we talk about sensation, we talk about awareness of stimuli. Whereas when we talk about perception, we refer to conscious interpretation of stimuli. So you can perceive only consciously some stimuli, okay? We don't talk about perception for those stimuli that are subconscious. Okay, so perception refers to interpretation of raw data in a cortex. Okay, so now the last part of today's presentation, we're going to talk about sensory coding. And when we talk about sensory coding, there are several principles we have to keep in mind. When a stimulus reaches a receptor, there are several events that happen. First of all, there is a reception of the signal. And so a specialized cell that is specialized for that kind of reception absorbs a physical energy. For instance, this can be light energy, right? It can be any kind of energy. That elicits a transduction pathway, which refers to this physical energy being converted into electrochemical energy. And because we know that ultimately here, what is any kind of physical energy that is picked by any sense, sensed specifically by a specific specific receptor is going to be converted in an actual potential. So we talk about electrochemical energy, okay? Bottom line, each receptor is specialized to absorb and transduce only one kind of energy. For instance, visual receptors in the retina are not activated by sound. They're only going to be activated by light. And the strength of the receptor potential determines how strongly the receptors are activated. Okay, so we're going to talk about receptor potential in a minute here. Okay, these are graded potentials. You know that an action potential is an all or none event, whereas a graded potential can vary in magnitude. Okay, so we're going to talk about receptor potentials as graded potentials here in a minute. So we're talking about sensory coding, which basically refers as how can organisms differentiate among stimuli or detect the strength of a signal. Okay, so that's the sensory coding. So receptors have to encode a certain stimulus, and they have to be able to gauge also the strength of this signal. And so sensory receptors and sensory neurons must encode 4 types of information that we can recognize: modality, location, intensity, and duration. What are these types of information? What do they refer to? Modality, which refers to the receptor physiology, refers to the fact that a specific form of energy can only be detected by specific receptors. Okay, and so basically, all sensory receptors are designed to respond best to specific modalities. Uh, and we have already talked about this because we have introduced the fact that specific receptors can respond to mechanic stimuli, thermal stimuli, chemical stimuli, photonic like light stimuli, pain stimuli, and pressure stimuli as well. Okay, so these is the modality. So the different forms of energy that specific receptors can respond to. Then we can talk about sorry, before talking about the next one, I want to talk about receptor potential, which I just introduced a couple of slides ago. Receptor potentials are also known as generator potentials. These are graded potential. Remember when we talked about difference between graded potentials and action potentials, we define graded potentials as potentials that can vary in magnitude. Okay, and in fact, receptor potentials vary. Their magnitude varies with the strength of the stimulus, and of course there is a threshold in stimulus amplitude that needs to be reached before response in terms of action potential is generated. And the amplitude of the receptor or generator potential saturates in response to intense stimuli. Okay, and so if a certain threshold is reached, the receptor potential can trigger an action potential. And this can happen in the same cell or in an adjacent cell. It depends on what kind of receptor we are talking about. Okay, are we talking about a simple receptor, or are we talking about, for instance, a special sense receptor, where we know that there is a separate cell that functions as a receptor, and then there is a sensory neuron uptaking the signal. Okay, whereas there are simple here the simple and complex receptors that we talked about. They are the very cells that uptake the stimulus. So in these cells, there is a generator potential that which is the receptor potential that in the same cell will elicit an action potential. All right. So with that said, now we're going to talk about location. Okay, we talk about location. This refers to receptive fields. So basically, when the receptive fields of specific afferent neurons code for stimulus location. For instance, we can give a very good example of tactile receptors in the skin. The smaller is the receptive field, the greater will be the precisions of the stimulus, which is known as acuity. And acuity, which again refers to the precisions of the stimulus, can differ, and it can be measured by a so-called two-point discrimination, which is the ability to perceive two fine points pressed against the skin as two different stimuli and not one. Okay, and so there are areas where the acuity is higher than other areas, and so the precision of the stimulus is different. Okay, so So in general, we can say that the smaller are the receptive fields, the smaller the distance between 2 points of stimulation that can be discriminated. Okay, and so areas with smaller receptive fields have better 2-point discrimination. What can be these areas? These are the areas where you have more you can distinguish precision of stimuli better. For instance, lips or fingerprints, it makes sense. Whereas other areas such as the back, the shoulder, these have worse 2-point discrimination than the areas such as lips or fingerprints, fingertips. It makes a lot of sense because for those areas you don't, the body doesn't need to have such acuity. It's not needed. You have to have acuity in the lips, in the fingerprints, these sort of areas where you need to discriminate much better between stimuli, and therefore there are smaller receptive fields. Okay, hopefully I've been clear. And so this 2-point discrimination is kind of an important concept. Okay, and again, the smaller the receptive field, the greater is the acuity. This is a principle that you have to remember for me. Now we talk about receptive fields. Okay, again, the smaller the receptive field, the greater the acuity. Okay, I'm repeating this because it's important. And here is an example to make you understand this concept. When we talk about the tactile acuity, this can be measured really by 2-point discrimination. And this is the ability to perceive 2 fine points pressed against the skin as 2 different stimuli and not one. And here there are these are the examples that represent 2 situations where in the first one is 1 signal that goes to the brain, and in the second one are 2 signals that go to the brain. And so what happens here is that secondary neuron is connected. One secondary neuron is connected with primary sensory neurons that come from certain receptive fields. Okay, here basically there are different secondary sensory neurons that are almost one-to-one connected with one certain receptive field. Okay, so this primary sensory neuron here, which is actually covering this field, is connected with one neuron. Okay, and in this other, is shown the green ones are the ones being stimulated, so it's 1 and 2 point discrimination. You see how in this case these 2 have dedicated secondary neurons, sensory neurons, that will send to the brain 2 separate messages. Okay, and so you have fewer primary neurons coverage here, and the fields are much smaller, and the 2 stimuli are sensed as separate. Whereas in this other situation, you have many primary sensory neurons that are converging on a single secondary sensory neuron, and therefore, no matter where you stimulate here, there is no discrimination, basically, discrimination between these 2 points that are being stimulated. All, no matter where you stimulate, you only have one signal that goes to the brain. Okay, so the 2 stimuli here are perceived as a single point. The 2 stimuli here, 1 and 2, here is a single point. The 2 stimuli here on the right are seen as 2 distinct points, 2 distinct stimuli. What are the areas that have this kind of innervation? What are the areas that are this kind of innervation? Of course, the areas where of your body, think about where you have more sensitivity, where you are able to discriminate better. These the 2-point discrimination are going to be your fingertips, your lips, whereas these are the areas of your back, your thigh, these kind of bulk areas where you don't necessarily have to have this very fine discrimination between stimuli. All right, so now talking about location, okay, we talked about receptive fields, we talked about the 2-point discrimination concept and the acuity. Now we still talking about location, we have to talk about, I'm sorry, my mouse is going crazy today. We have to talk about lateral inhibition. And so this is a phenomenon that probably you can think about, you can recognize. And it occurs when a strong stimulus, which is applied to the receptive field of a neuron, causes the inhibition of neighboring receptive fields, okay? And so imagine that you are putting a pin on a certain area of the skin. What happens is that if you are, especially if the stimulus here is pretty strong, so it's pressing pretty strongly, what happens is that the stimulation of this a neuron, the secondary neuron, will determine, because of lateral connection, will determine the inhibition of the neighboring secondary neurons, okay? And so, which means basically this cannot be fired. Even if you apply a stimulus, because there is being inhibition, you cannot feel basically anything around that area, okay, where that is being intensely stimulated. All right, so I think I said everything to you. I was trying to remember if there was anything else that I wanted to tell you. Okay, so this is we're still talking about location here. Okay, so we talked about location just now. We had talked about modality as a first. This should be highlighted, sorry. It should be highlighted in yellow, okay? Talked about location. Now we're going to talk about stimulus intensity. What happens when you intensify a stimulus, when you make it stronger? Okay, if you put more pressure rather than less pressure, for instance, on skin, this will produce a higher frequency of action potentials, as shown here. So this represents, over here represents stimuli. This is a stimulus that is less strong than this one, right? And you see how the resulting action potentials here, okay, are less or more frequent, okay? So a lower or less strong stimulus produces action potentials that are of less frequency, okay? Or vice versa, a higher stimulus, a stronger stimulus, which of course will result in a higher receptor potential, because there is always this graded potential that needs to be initiated, of course, before an action potential an axon potential or action potential, right, can be stimulated, okay? So axon potential here is the action potential. It's calling it axon potential because you know that action potentials are initiated in axon hillock, right, of axons. So you can call them axon potentials as well. It's not very common, but sometimes they call them axon potentials other than action potentials. And so a stronger stimulus determines a stronger receptor potential, which will result in higher frequency. Remember that action potentials are all or none events. You cannot change the intensity of an action potential. Is that right? Okay, we talked about this extensively. Please, this is a very important concept. You cannot you absolutely cannot get confused on this one. Graded potentials can vary in intensity. See how this receptor potential, which is a graded potential, is lower here than here, okay? But an action potential is always the same magnitude, right, of plus 30 millivolt. It does not change. See, the magnitude is over here. You don't see that they go higher than this, okay, no matter what. Okay, its action potential is the same intensity here as here, even if the stimulus is less strong in this situation, right? This is the stimulus that determines this action potential. This is the stimulus that is determining these ones. So the intensity of action potentials or axon potentials does not change. What changes though is the frequency, okay? The frequency. So this is very important, okay? What is the result in terms of action potentials of increasing stimulus intensity is the frequency of the action potentials, not the intensity. So if you have questions in the test, do not get confused, okay? It can be tricky, okay? It can be true or false questions that say, for instance, a stronger stimulus in a receptor will determine a higher or stronger value of an action potential. It's false. It's the frequency, okay? It's not the strength, because action potentials are all or none events. They never change in intensity. And this is something we talked about extensively when we talked about action potentials. Okay, the fourth point here we're going to talk about is the stimulus duration. And when we talk about stimulus duration, of course, what it's the length that a stimulus is given, the length of time that a stimulus persists. For instance, I can do a touch for longer time or for shorter time. And so there are different types of receptors that we can talk about in terms of how they respond to the duration of a stimulus. And these are the tonic receptors and the phasic receptors. The tonic receptors show little adaptation in response to a prolonged stimulus. And so this is your receptor here, okay? There is going to be your receptor potential, okay, which is going to elicit an action potential, right? And look at the situation here in the first the tonic receptor. So you have a prolonged stimulus. Now, the stimulus, the duration of the stimulus is the exact same in the two examples given here in the left and in the right. What it wants to point out to you here is the different types of receptors that exist, which are basically two types, in response to a prolonged stimulus. There are the tonic receptors that show pretty much they're refractory to adaptation to a prolonged stimulus, meaning that the receptor potential is pretty much active for the duration of the stimulus. You see, it's there. It doesn't go down until the stimulus goes down pretty much, okay? And this results in, of course, several action potentials here, which go on kind of like in a pretty much steady frequency for the duration of the stimulus. Now let's look at the phasic receptors. The phasic receptors respond differently because basically their receptor potential there is a receptor potential that, of course, is initiated upon the beginning of the stimulus over here, but it goes down right away, okay? And then what happens is that there is another one that fires right at the end when the stimulus pretty much turns off. So there is really one in the beginning, one at the end. And if we look at the action potentials that are generated, there are action potentials that are generated in consequence of receptor potential at the beginning and at the end. Okay, so it's a pretty different situation here and here. Okay, and so we say that the phasic receptors adapt quickly. They have already adapted to the stimulus. They're not responding in this time. During this time of the stimulus duration, there is no response. So there has been a quick, rapid adaptation, okay, for the phasic receptors, whereas the tonic receptors show little or no adaptation to a prolonged stimulus. All right, so pretty much that's all I have, okay? So if you can go over this lecture for me, these concepts really that we have talked about, conscious versus unconscious perception, general and special senses, shouldn't be very hard for you to grasp. Remember, the special senses are 5. If you remember the special senses, then you can remember the generalist one, because anything that is not special is general, right? And sensory neuron classification by type, stimulus detected and location, and the concept of sensory coding that we just talked about. Okay.